Now we would like to get started with this evening's program. So I welcome Frank to the stage. Hi, Frank.
Hey, thanks for the introduction, Emma. As Emma said, my name is Frank Santana. I'm the collections manager in the herpetology department here at the NAT. Happy to be here sharing a behind the scenes tour. Like Emma said, this is kind of a new format and it's hard to squeeze everyone in for in-person tours. And so for you to get a virtual tour, it's pretty exciting for you to see our collection and talk about our work both in the lab and the field.
Absolutely. So Frank, you mentioned that you are the herpetology collection manager, which is kind of a mouthful. A lot of new words in there. Can you start by defining herpetology for us?
Sure. So herpetology is the study of reptiles and amphibians. Reptiles and amphibians are grouped together in a field of study, even though they're pretty different from each other. So reptiles are scaled animals. So they have scales that protect their skin from physical abrasion. It also protects their skin from like evaporation of water, so they can live in environments that are hot and dry, compared to amphibians who don't have scales, and their skin is really permeable. So they don't have scales, their water evaporates through their skin pretty easily. And so, for example, if you kind of think of a amphibian skin, when we're working with chemicals like bleach and things like that, our skin is a little bit permeable, but it's not, completely super permeable. If you spill a little bleach on your hands, wash it off quickly and you'll be fine, right? But if you're working with bleach, and you should wear goggles when you're working with bleach, because if you got some bleach in your eye, you can kind of imagine your eyes being similar to the permeability of an amphibian skin, right? So you want to protect your eyes from getting bleach in there. So for an amphibian, if they got bleach on their skin, it would be really detrimental for them. So amphibians are kind of like canaries in the coal mine. They're indicators of the environmental quality and the habitat in which they occur in. So if there's any problems with water quality and things like that, amphibians are really sensitive. And kind of going back to thinking about why reptiles and amphibians are studied together, though, way back in the 1850s, Carl Linnaeus was a biologist that decided to thought about how to organize animals all throughout the world on planet Earth in different ways. And he kind of grouped all reptiles and amphibians as creeping animals. So herpetology, the root of that word, means like creeping animals. And so they got lumped together, even though they're a little bit different from each other.
Yeah, kind of a bit of a mistake in taxonomy there. Carl Linnaeus made a lot of mistakes. You can certainly do some research into the science he did about people. He tried to create a scientific basis for racism, and he got that all wrong as well, of course. So creeping animals doesn't sound great, but I know you love them, Frank. Can you tell us a little bit about how you got so interested in reptiles and amphibians?
Yeah, so I grew up in San Diego County, and I was lucky enough when I was younger to be able to run them out around my yard and look for animals, and my mom would just let us run around. And I remember specifically the first time I had seen an amphibian was I lifted over like a cylinder brick, and under it, it was really moist in there. And there was a organism that was really small, slender, and it looked really like a worm. But then it had these tiny, tiny little feet and these little legs and arms, so little legs. And I didn't know what it was. And then I knew it was something different. And then looking back on it now, I know that it's a slender salamander that I was able to find in my own backyard. So that was really cool. I was always interested in microorganisms and wildlife just in my own backyard. And my parents weren't really into camping or hiking or nature or anything, but luckily my uncle was really into it. And he took me and my brother and sister on our first camping trip in Anza Desert State Park. I remember driving along the roads at night looking for snakes. We found a snake on the side of the road. We were able to get out and check it out. I kind of been interested in reptiles and amphibians ever since then.
That's awesome. And how did you translate that interest into working at the NAT?
So I really didn't know what I wanted to do when I was in high school and then college. I dabbled in like computer science and things like that. But then I was lucky enough when I was in college to be able to take a herpetology class. I knew I was interested in it, but didn't really realize you could do it as a career. And then from that, I started getting a real more academic and rigorous and scientific interest in it. And I just decided, yeah, I want to be a biologist that focuses on reptiles and amphibians. And after I graduated from college, I went to school in Santa Cruz, and then I came back here to San Diego. I was living with my mom in El Cajon and kind of just figuring out what I wanted to do with my life. And she saw an ad for a volunteer in the herpetology department at the Nat. And so I went in and interviewed for that and was able to volunteer. And that really got my foot in the door. I got some more professional experience outside of the Nat, working at the San Diego Zoo for 9 years, and then dabbled in teaching at community college, which is a really cool way to improve my science communication skills. And then a job opening at the Nat a couple of years ago, and I've been here since then.
Sweet. And now you are the Nat's Herpetology Collections Manager.
Mm-hmm.
So you defined herpetology for us already, but what exactly is a collection? Briefly, we'll show everyone in just a second. And what does it mean briefly to be a collections manager?
Yeah, so as you said, we'll kind of give a virtual tour of the collection that we have, but in museum science, a collection is any set of objects that's used for research and study. So at the Nat, we have a lot of different departments. We have ornithology, we have mammals. So every department's gonna have different taxonomy, different groups. We even have a research library. We have some really cool books. So even the library books are considered a collection. And then herpetology, of course, we have reptiles and amphibians in our collection. And they are, just a collection of biological specimens that are used for research. And we'll get into some more details of how specific examples of how those specimens are used and whatnot.
Thank you, Frank. Sounds great. We'll go ahead and follow you down to the basement now and check out that collection. So here's our video. Let me know if you can't hear it. Where are we, Frank?
We're down in the herpetology wet collection, and this is where I spend a lot of my time working on our collection research. And we have a total of 68,000 specimens preserved in ethanol down here.
So they're called wet because they're preserved in ethanol. That kind of begs the question, do we also have a dry collection?
Yeah, so the wet collection is everything where they're going to be preserved in the liquid ethanol, but the dry collection is our ossified or bone collection. So upstairs we have 8,000 specimens that are bones and skins of specimens.
So you're using this word specimen a lot. Can you tell us a little bit about what that means?
Sure, so specimen is anything in the museum that we have detailed records on. So in the library, the specimens are books. And here are specimens, of course, because we're herpetologists, we're working with reptiles and amphibians.
Very cool. Can you tell us a little bit about the history of these specimens, the history of the wet collection?
Sure, so the collection in herpetology really started growing in the 1920s when the curator back then, Lawrence Klauber, collected a lot of reptiles, and his focus was really on rattlesnakes. So you can see behind me we have a lot of rattlesnakes that a lot of them were collected by Lawrence Klauber in the 1920s and 30s and 40s. And he really jump-started our collection. So now we have a total of 9,000 rattlesnake species, which is the largest, not species, 9,000 individual rattlesnakes, and it's the largest number of rattlesnakes in any museum collection in the world. So people come all over to do research with these rattlesnakes.
So we see that that snake is preserved in ethanol. Can you tell us a little more about what ethanol is, and also, isn't it like very, very flammable?
So ethanol is essentially an alcohol, just like you have like a bottled wild turkey at home, maybe, right? It's really active. If you spilled some of that on your hand, you'd feel it kind of evaporating. And so the ethanol is very, very active. So if there was any spark or any problem that caused a fire down here, it would be a really, really big problem. So we're in the very, very basement of the Natural History Museum, and you can see there's some fire suppression plumbing up here, so these red pipes, and they have special foam that would put out a fire really quickly. And unfortunately, it went off accidentally a couple years ago, so we had a test of it, and we know it works, but we don't want it to go off unless it's really necessary. And then we also have some features on the bottom here. So our floor, it's really crickety, and it's kind of like this false bottom. So we're not really standing on a solid floor right now. It's actually hollow. So in case there was an earthquake or another disaster and the jars spilt that ethanol, it would all run to a kind of low point on the floor, and we could drain it out really quickly.
Great. So a lot goes into keeping the collection safe then.
Yeah, there's a lot of work to maintain it. We're constantly checking systems, keeping the temperature controlled to limit risk of fire. There's a lot of work that goes into collecting, maintaining our collection here.
Awesome. Thank you, Frank.
You're welcome.
All right. So that was awesome. A great introduction to the collection. We do have a comment in our Q&A box. Pickled snakes in ethanol. The fire marshal must hate that collection. You were probably right, Charles.
So that space was engineered really to conform to really high safety standards. So it's at the very, very bottom of the building. It was finished in 1999, so it's more modern than a lot of the older parts of the museum. Keeping it at really cold temperatures is it's always been a challenge, but we maintain it at cold temperatures also to reduce the flammability. So it can get pretty chilly in there sometimes.
Thanks, Frank. And then another question in from Noah. He asks, are there any reptiles now extinct in your specimen collection?
We have some species that are locally extinct. So they used to be found in Santana County, and now they're not there anymore. I can think of amphibians that fit that description, but not reptiles off the top of my head. But yeah, it's one of the benefits of having that collection is it has a really solid record, right? Rather than having someone's notebook where they described, oh, it was a snake this long and it looked like that. By having a physical voucher or verification, we could say that species did occur at that spot.
Great, thank you. So that's some of the significance of a collection. We have one more question from Elliot. Are you going to show any of your pets? And Elliot, if you mean pets in jars, then yes, absolutely. If you mean Frank's pets behind him, we may not have time for that, unfortunately. So,
I leave the dead pets at work and the few live pets at home, but yeah.
So Frank, you mentioned that there's 68,000 specimens in the collection and that some of those have been in there for a really long time. Can you talk a little bit about how a specimen ends up in the collection though? What does it mean to collect?
Yeah, so in the early 1900s when our collection was growing, most specimens were collected as part of the expedition where it was really focused, and the goal was to collect maybe a relatively large amount or large number of specimens and add them. These days, our collecting is a little more limited, so we do still do expeditions and we collect specimens from those expeditions, but we also take a lot of photo vouchers, so we don't do as much preservation and collection from our expeditions these days. But we still get a lot of specimens that are brought to us from research projects where if a researcher sees a roadkill animal and we have a good relationship with them, they'll bring it into us and we can add it to our collection so it doesn't get wasted, it gets used for science. And we also have similar relationship with the public. So we have a good connection with the Project Wildlife, people at Project Wildlife. And if you saw a cat that had gotten a hold of a snake and the snake is still alive and you bring it to Project Wildlife, If that's, unfortunately, if that snake doesn't make it, then we can make some use of it for scientific research.
Thanks, Frank. And once you have that snake in your possession, what are sort of the next steps? What happens to it between you obtaining it and it ending up in the collection?
Yeah, so there's some really key and vital sets of information that we need, which are pretty basic. We just need the person who collected it, the date they collected it, where they collected it from. So it could be even like a cross street or something. But the way we have our phones now, you can even just take a photo and get a coordinate off of your phone from the GPS in your phone. But knowing where, when, and who, that really gives us a solid record. And then we store that animal in the freezer for a while until we have time to process it. And then we do preparation of that specimen where we inject them with formalin. So formalin is a preservative. It's also a mutagen. So we have to be really careful and use that in the fume hood. We'll show a video of the fume hood in a little bit. And then put it in ethanol for long-term preservation.
So as someone who obviously really loves and cares about reptiles and amphibians, I know you're not just going through all that for fun. It is not like my rock collection at home. Why collect? Why do you guys have all those jars down there?
Yeah, so I think it was Noah that asked the question about extinct animals that were once found in San Diego. So that's an important part of it, is that even if animals aren't extinct, they're declining because of human impacts, right? So having a record of where things used to occur and thinking about if there's any effort to restore ecosystem, and thinking about what species might want to go back in that area, or also thinking about projecting future impacts on wildlife as our human footprint expands. And some other examples include kind of thinking about the history of the taxonomy of the species. So having those records and having a large number of specimens allows us to do research in the future to try to understand more about what is a species and what's different. Lots of times the identification of species changes over time. So having those records help us also identify changes in species definitions. Yeah.
Cool, makes sense. So we're actually going to check out a couple specific examples of some of Frank's favorite specimens that have some really important research and conservation implications. We've got a few more questions in the Q&A box that are really great. A couple of them we're actually going to answer a little bit later through one of the videos, and the ones about Lawrence Clobber we'll go ahead and save for the end of the program tonight. All right, so I'm going to go ahead and share one of the collection specimens.
All right, let's check out some red-legged frogs and bullfrogs over here.
So Frank, now that's a little quieter, can you remind us what specimens we're seeing?
Yeah, so this is a California red-legged frog, and these ones were actually collected in the 30s and 40s, and it's an important record for San Diego County because they're no longer in the wild here in San Diego County. They're locally extinct. We're working on projects to reintroduce them back in the wild, but essentially it's just like a very small population right now that we've reintroduced. And having this record really provides a lot of research opportunities. So frogs throughout the world have been affected by this fungus called the chytrid fungus. So this fungus has kind of spread throughout in the 80s and 90s, but it wasn't even documented as an official disease until 1999. So the frogs were dying off, and it was kind of like this mystery of why they were dying off. And then having this record, we can actually go back and swab preserved museum specimens for that fungus now. And that's important because it helps us understand the patterns and the causes of that extinction of whatever species you're swabbing or whatever populations. So by knowing what caused that decline or extinction, we can address future reintroduction efforts. So if it was a lot of chytrid fungus, maybe we would treat those frogs before we reintroduce them back in the wild, or monitor for chytrid really closely if we ever do any reintroductions in certain areas.
Very cool. And I think you mentioned that there's another
threat to the species as well. Can you talk a little about that?
Yeah, so the fungus is one of many threats to native amphibians here in Southern California and throughout Baja California. But another big problem for all species throughout the world is invasive species, right? So there's invasive plant species that will really spread and take over. And then for the red-legged frog specifically, a big problem is the bullfrog. So bullfrogs are not native to California, but they've taken over. So if you go to mission trails and go hike at night on a rainy night, you might find some bullfrogs in the San Diego River. They're not supposed to be there. They're kind of cute when they're small, but they get really big. This frog will gobble down a red-legged frog so easily. I've actually seen one with a rat in his mouth, a native rat. They'll eat birds. So it's a real big problem. And so if we're thinking about reintroduction and having this record here of the bullfrog, having the record of where they occurred at certain times, it lets us know that they're also contributing to the declines of the native species in those areas. So by tracking, kind of keeping that history of where the bullfrogs were, we know when the declines were, being impacted by bullfrogs. So this one is from Riverside County. Again, that doesn't belong there, and it's actually from one of the nature reserves out there. So by knowing where it is and when it was there, we can start to piece together the requirements to restore that habitat.
So you said they've been here a pretty long time. Why were they introduced originally?
They were actually brought over when the red-legged frogs started declining. So the red-legged frog is the largest native frog in California, and they started declining because they were being eaten for frog legs. And so it was a good source of protein. And so mostly in the Bay Area in San Francisco for the 49ers, they were eating red-legged frogs up there during the gold rush. And then so they started declining so much that people brought in bullfrogs as a substitute for food. And then from there, they just kind of spread all over.
Wow. Thank you, Frank.
All right. So pat yourself on the back if you guessed that bullfrogs have been around here for about 170 years. Also, let us know in the chat, just because I'm curious if you've eaten frog. I have not, and I'd like to. I'd like to eat a bullfrog. Frank, if you catch one out in the field, please bring it back to the museum for lunch.
We'll have a barbecue.
Perfect. So clearly you learned a lot from the collection. You learned about the red-legged frogs and the bullfrogs as well, but does that research have any real life conservation implications? What have you done since learning all of this?
Yeah, so knowing where the bullfrogs are occurring and thinking about possible reintroduction sites. So as I mentioned in that video that you guys just saw, we are collaborating with some great partners on a California red-legged frog reintroduction and conservation program. And our partners at the USGS are kind of leading the work in the United States, and they put a lot of effort, a lot of time in removing bullfrogs from the reintroduction sites. And we have some property owners at one of the sites, Judy Wheatley and Chuck Wheatley, who also helped facilitate that process of removing bullfrogs from the field site where we introduced the red-legged frogs. So it's really important to make sure those bullfrogs aren't there before we can work on the reintroduction and get the red-legged frogs in. So we've done reintroductions for 2 years in a row now, and it's a slow start, but it's going good so far, and we're hopeful for increased success in the future. And also thinking about the populations in Mexico, it's a bi-national project, and we have some great partners down in Baja California, Mexico, specifically Fauna del Noroeste, and it's also known as FANO, and that group is led by some great Mexican biologists down there, and they really know the land down there. They've done surveys to see which sites might support red-legged frogs and where the bullfrogs are. And there are 10 sites that are known for where red-legged frogs occur across 10 different sites. And so those sites where they exist, bullfrogs don't exist. So luckily, we have been able to identify those sites, and we're working to improve the habitat at those sites without the bullfrogs.
Cool. We've got a couple of videos of you all at work along with your partners. Can you see this? I don't know what I'm doing with screen share right now.
Yeah, it looks, the video's up, but it's not playing yet.
Tipo de acciones son comunes.
There it goes. So we got some funding through the museum to collaborate with our partners down there at Fano to build these frog ponds. And you can see it takes a lot of heavy machinery to build these ponds. And the ponds are built adjacent to a stream channel, far away enough so that when there's like a really heavy rain, it won't damage the habitat. And you could see after those ponds were built, immediately they started filling up with groundwater from the bottom. And luckily there's no bullfrogs out there. So we're working to restore and improve these habitats in Baja California, Mexico, so that we can have better outcomes and increase the population sizes of red-legged frogs in Baja California. And of course, when you build these ponds, they've been there for how long? Almost going on four years now. And they start to fill in with cattails. So the red-legged frogs prefer the open kind of habitats. And the open habitat really helps them to be able to lay their eggs, to do normal breeding behavior. So we have to go down every year and manually pull out the cattails. It's a lot of work. You can see one of our volunteers, Roberto, there is really celebrating that he removed that cattail out. We did it for one full day, and it was kind of exhausting, but really rewarding when you pop out a cattail. So we're removing most of the cattails to help the red-legged frogs.
Awesome.
And we've seen some good outcomes so far. So the first year that we did a focus survey for eggs, this is us looking for egg masses in the streams and creeks and ponds. We found 20 egg masses in the ponds at the site here that we're showing. And the next year we found close to 40 egg masses the next year. It's a cool video of some of the egg masses. And it's really exciting when we find egg masses because it's like, you know, they're so pretty. I mean, look at that. It's just like, it's this like diamond in the rough that, you know, we're so excited to see this healthy reproduction, signs of healthy reproduction.
Very, very exciting news. This is awesome. We've got a question from Elizabeth about frogs. So Elizabeth asks, it's cool researchers are able to test for fungi on frogs in that historic specimens. Are they also able to test for chemicals like pesticides?
That's a good question. I'm not familiar with any research that has done that. I think the challenge with that is whether or not the products break down during the preparation and preservation. If they might be too broken down to be able to track them, but it's possible, something to look into. And but we haven't been approached, as far as I know, by any researchers who are looking at that. But that's one of those things where, you know, maybe in the future there'll be some technology where you can look for traces of pesticides to try to see how population was affected. So our collection is kind of like this thing that we hang on to it, and we know there's some research value there, but we don't know what kind of technologies will come up 20 years from now that might be able to answer some really cool questions that we couldn't even imagined back in our 2020s.
Just like Lawrence Klauber couldn't imagine that, you know, today you'd be doing the things that you're doing. It's pretty cool to think about that legacy.
Yeah.
We'll talk a little bit later about some of the really cool tech that's coming out now that Frank has participated in. One more question about frogs before we move on. Noah asked, what would you do if bullfrogs invade the artificial pond?
So if the bullfrogs invade the artificial pond, we can physically remove them. So capture them, kill them. Our partners will use guns to shoot the bullfrogs and just try to eradicate them, remove egg masses. Another option might be to have to drain the pond. So bullfrogs can't survive without permanent water. So if you drain the ponds and it's a dry year, maybe that could also get rid of the bullfrogs. But luckily so far, the populations seem pretty well maintained because they're pretty far away from population centers. So in San Diego County, the problem is that there's all these people's backyards that have ponds. If you think about golf courses, there's bullfrogs in all those golf courses. So it's really hard to eradicate them now. So the further you get away from people, the better. So that's it unfortunately limits to where we can do restoration in a practical sense. Good question though.
Yeah, that's a great question. So we're gonna move to another specimen with different research and conservation implications. And this one is very seasonally appropriate. So we're excited to share it with you.
So, Frank, what you got in here? A bunch of skeletons in your closet?
Well, no skeletons, but I have some really interesting specimens I want to share with you. This is our holotype cabinet, where we keep our specimens that are representative of an entire species. So a holotype is any specimen that, for example, represents everything that is of that species. So if you were found a snake out in the wild and you weren't sure what it was, if you thought it was, maybe between two species, you could bring that snake in or take some really detailed photos and compare it against the holotypes that we have in our collection. So how many scales it has, how big it is, the width and things like that, and the patterning are all going to be compared against that holotype. And we have our holotype cabinet here, and it's really important that we keep these holotypes protected because they're really a way to document what species we have throughout the world. And so we have them under lock and key here. Let me unlock it for you and we'll take a peek.
Cool, so no skeletons. It is the wet collection after all, but you do actually have something with some Day of the Dead connections in here, right?
Yeah, so I'll tell you all that story of how it relates to Day of the Dead. We have all of our holotypes here. And then we have a rattlesnake here in our holotype collection. And this rattlesnake was collected in the 1930s. And Lawrence Klauber, who was the curator who we talked about in that time, who was really, really into rattlesnakes, he would have friends from all throughout the world send him rattlesnakes that he would add to the collection. And so this was added in 1934. And when he collected it, it was previously described as a different species than it currently is known as today. So it was on our shelves with all the other specimens, not under lock and key, just our general collection. And then we discovered that, hey, this thing is actually a holotype. So how did it become a holotype? It was just sitting on the shelf, and now it's a holotype. What happened was scientists who were out in the field that study rattlesnakes in the area where this rattlesnake was found, which is in the Yucatan of Mexico, were looking at the specimens and catching animals in the wild and determining that, wow, there's something going on here. There's this kind of pattern of this rattlesnake that maybe is different than the rattlesnakes we see around it. And so what they did was they collected a lot of data on rattlesnakes in the wild, and they came in and looked at our collection. This was all happening during the pandemic, so they looked at really detailed photos and asked to look at this individual specimen and determined that this is actually a new species of rattlesnake. So it's not what we thought it was when we first collected it, when Klauber collected it, but now we realize that, as of 2020, when this paper was published and these researchers did their work, they discovered that, this snake has different scales, patterning on its body. It's a different size than the other rattlesnake species that we thought it was before. And so they got to name that rattlesnake. So it's a new name. It's only been around for a couple years now. And it is Crotalus for the scientific name. That's the genus. And a lot of rattlesnakes are in that genus Crotalus. And the species is Mictlantecutli, which is an Aztec word for god of the underworld. So this is the rattlesnake of the dead. So for thinking about Dia de los Muertos, this would be a great rattlesnake to have on your altar to call back your loved ones, because it's the rattlesnake of the dead from Yucatan, Mexico. It's really exciting that we have this in our collection because it was just this cryptic species hiding under our noses the whole time.
Very cool. So cryptic species, distinguishing one species from a very similar looking one, is this just splitting hairs? Are there any, real world consequences to that?
Yeah, so if you kind of think about, kind of labeling things and grouping them into different species, it has real conservation implications. So if you look at that photo there, that's the specimen, that's the holotype for that Corylus mictlan. And before it was grouped in with a larger group of more widely distributed species of rattlesnake. So now we know that this is actually a different species. It occurs in a smaller geographic area. So knowing for conservation purposes that this is actually different, it's unique, it occurs in a smaller geographic area. If you're managing the populations of wildlife for this species, now you want to kind of like think about it from a different perspective. With that smaller distribution, you have to worry about different threats that might be affecting it. Maybe the species has a different natural history where one's nocturnal, maybe one's active during the day, diurnal. So those would have different management implications for protecting it. So calling it one species or another really has like real world conservation implications for them.
Makes a lot of sense. So you mentioned that researchers came in, they made this discovery, and I have to admit, when you first told me that, I pictured like they were just looking in the jar very closely. Maybe they had a magnifying glass, and there's a couple other people in the Q&A box who have similar questions about how researchers access the specimens and what they can do with them. So we'll go ahead and answer that with our next video. Frank is going to show us around the herpetology lab.
All right, so where are we now, Frank?
We're in the lab right next to the wet collection, and this is where we do a lot of our prep work for the specimens that we've shown you already and talked about. So this fume hood is our dedicated fume hood right here where we prepare our specimens. So you can see there's a lot that we already have like being prepared right now. And it's just our dedicated space where we can safely use formalin and inject the specimens with formalin and then eventually add the ethanol for the long-term preservation.
And what else do you do in this space?
We also do our computer work down here. We have a lot of molecular biology equipment, and we also have like our freezers to preserve specimens before we've had time to prep them. In addition to having some photography equipment and printing equipment down here too.
Awesome. And we're in here because you're going to give us an up-close look at one of...
I'm sorry, we should warn you. This specimen is a little more gruesome than some of the previous. There's no blood, but consider this your heads up that this one is a little bit, I don't want to give it away, a little more Halloween-y, if you will.
The coolest, Halloweeniest specimens in the whole collection, right?
Yeah, so I have some California king snakes from our collection. This is a local species that you can find here in San Diego County. And as we talked about earlier, when researchers come in and want to do different studies with the specimens, they will come into our lab and they'll pull a jar of specimens that they're interested in and do different measurements on them. So we'll kind of go through an example here with the California king snake. And we had a recent researcher come in and he published a paper in 2020 looking at the diet of California king snakes. So I'll remove some of these for you and give you a little example. So you can see when there's a study where someone's interested in the diet, they can actually open the specimen up and look at what they had in their guts to see what was in their stomach at the time that they were preserved. And that can provide a lot of information on what the specimens were eating. And so oftentimes you have to make an incision, you kind of have to do some destructive sampling where you kind of like alter the specimen. But in one case we were really lucky because that didn't need to happen in terms of like what this animal was eating. We got some information just from looking at the exterior of it. So this California king snake actually found a horned lizard in the wild and constricted it, squeezed it, and killed it, and then decided, of course, it was its prey item, so it wanted to eat it. Unfortunately, the horned lizard was too big for the king snake, and it punctured through the king snake's head. You can kind of see there's a little bit of where the puncture wound came through on the top here. So it was a bad day for both of them because the king snake thought he was going to get a nice meal out of it, but it didn't work out so well for the king snake. Yeah, so this is a really rare event when you're out hiking. You know, it's rare to see a predator catching a prey item, but we kind of have this record for time immemorial of the king snake eating the horned lizard.
That is gnarly. Thanks, Frank.
All right. Thank you, Frank.
Yes, that's a fun example of how our collection is used. These images here show different snakes, a couple of king snakes and a rattlesnake that were opened up to look at the anatomy or biology of these specimens. So things like diet, you can look at different organs. So people look at different tissues to see what the animal was like when it was alive.
Yeah, so Tom, here are some photos of those snakes that have been cut into. Thanks for that question. And I swear those aren't plants that are continually giving us the next question we're going to answer. So that was awesome. I know that you don't spend all your time though in the collection looking at cool things in jars. Sometimes you're stuck behind a computer like the rest of us. So do you mind showing us a little bit of what you do outside of the collection to track these populations?
Sure. So we have a really great resource that we put together, which is the Amphibian Reptile Atlas, also known as the Herp Atlas. And I think there's gonna be a link in the chat posted if you want to check it out yourself. And it's a cool way to, I'm gonna share my screen now.
Yeah, and I am way behind on sharing links out with you all. So you're about to get spammed while Frank shows you this website.
How's that looking, Emma? Okay, so if you look at this website on the screen I'm sharing, this is our Herp Atlas, which is a great resource for all the reptiles and amphibians that we have in our collection, and then some. So if you look at these tiles here, you can click on any of these species. Let's do flat-tailed horned lizard. Click on that one. And then a page comes up that has a little bit of an overview of their natural history. It shows a map. You can see on the cursor here a map, and you can click on map it and see detailed information about all of the flat-tailed horned lizards that we have in our museum collection. So it's a great way to really start to, you know, instead of looking at a spreadsheet and looking at a bunch of numbers, you know, seeing a geographic map helps you kind of like understand the patterns of where those organisms occur. And so let's do an example here. We'll go back home to the home page, and let's look up for some information about those California king snakes again. So you can type in California king snake, there it is, and then go. All right, so we have the California king snake. So I'm going to click on the information for that species. Scientific name is up here, Lampropeltis californiae. Natural history, some good photos, some facts about it. And then let's go to the main feature, which is mapping the collection. So you can see this map is loading. It's pretty cool. It shows you the outlined area is showing the distribution of the California king snake. So you can see it's kind of a misnomer because it's found outside of California, but it's just a species that occurs in the West in a variety of types of habitats, deserts, scrub, chaparral habitats. Okay. So all of those red kind of balloon icons are records from our actual collection. So it's kind of cool because you can see, let's zoom in on a couple of them. You can see we have records from all over the place. And let's zoom in a little bit more on San Diego County. I know there was someone on the chat that said they were tuning in from La Mesa. So let's see if there's any California king snakes in La Mesa that we have in our collection. Okay, where is La Mesa? La Mesa is, okay, around here, right? So you can click on one of these and then it shows you information for that record. This one's actually from El Cajon, my hometown, and it was collected on August 3rd, 1946. If you click on this beautiful record, you can see photos and information that has more details about that record. Okay, so these are the specimens that we have from our collection. If you click on another one, they're all they're usually older. So 1929, as I said, we bring in less animals these days, 1930. So this is the kind of historic distribution, but of course the collection is also used for thinking about current patterns and current problems with sustaining biodiversity and wildlife. So what's cool is that we also have a project on iNaturalist. If you click on this rascals here, it shows all these blue icons now. So I'm gonna get rid of the historic museum ones, or let's leave them up for a minute. So you can see that all those blue balloon icons represent records from iNaturalist. So these aren't.
I just had a question from
Oh,
Teresa.
yeah.
Is there an app or place we can upload a photo for identification?
Yeah, there's the iNaturalist app, which is a mobile app that you can get on your phone. And you can, if you're out hiking somewhere and you see a snake and you feel safe and you're not gonna get injured, you're sure it's not a rattlesnake and whatnot, or a lizard, any insect, any animal that you want to see, plants even, you can take a picture of it and upload it to iNaturalist and it creates this record. For reptiles and amphibians to show up on this map, we're only going to look at those, and you have to join a project on iNaturalist called Rascals. And so there's links that I think Emma's going to post at some point, and we can also send out if you have any questions and specifics. There's also some YouTube videos that we have like trainings on iNaturalist that you can go back and look. It's a really cool resource because you can see this one, whoever the observer Biohex is, he or she put some cool pictures of it, right? So it's a really cool record. And let's get rid of the historic and let's just go to the iNaturalist observations. So the balloon ones are exact locations. The blue dots, you can see there's one out in the ocean over here. That's kind of like an obscured record. So some people just don't want to say exactly where they saw something, so they obscure it. You can do that if you want. And then as researchers, we can ask for permission to see this data too. So even if you obscure it, we can get permission from the user to get records. So it's important for current research. And again, comparing this to where they used to occur to where they occur now, you can see there's some specimen records out in more rural areas. There's not as many in developed areas. But so let's check out this record. It's kind of interesting. It's in the college area where there's a lot of suburban development. And you click on that and it shows this. So it's a roadkill specimen, but it was observed in 2021. So it's a recent observation. And from personal experience and from knowing a lot about this species, California king snakes actually do well in urban and developed areas because they are generalists. They eat all types of different prey items. They eat birds, they eat mice, they eat lizards, they eat other snakes, they eat rattlesnakes. So they do pretty well in urban areas. So, but having this record, even if it's a roadkill one, lets us know, okay, hey, there's a population that exists in 2021 in the college area. So kind of managing how we want to maintain biodiversity in these kind of urban areas, including canyons that are kind of like these fingers of wildlife where wildlife can survive surrounded by development. And our museum here at the Nat has a project on canyons. So it's cool to think about it that way.
Very cool. And so obviously, all of these observations that you make on iNaturalist are really useful to scientists, to researchers in their research and in their conservation. As an example, I'm in the education department, and at one point, our campers actually got some observations of green anoles in Balboa Park, those little really bright green lizards. It turns out they had escaped from the zoo and established a population in Balboa Park. And that information came from, I think, elementary students, but ended up being really useful to our scientists because it showed how those species had moved. But I'll also say there are some great, more selfish reasons to use iNaturalist. When you're, out for a hike or in your backyard, like Teresa was, you can take a photo of something you see, something you're curious about, and it has really, really good AI that suggests what you're seeing. So it will tell you the plant or the insect or the reptile, and it's typically correct. If it's not, a scientist or a community scientist, which is a person who just knows a lot about nature, will come on and correct it for you and let you know what it is that you saw. So it's really a super fun app, and you can use either the Herp Atlas or iNaturalist as well to get an idea of what you might want to keep a lookout for when you're going somewhere new. Here's a
Yes, it's a great example. So here's a picture of a green anole from Balboa Park.
It's pretty cute, but it shouldn't be there.
Mm-hmm.
That's very cute. Cool. So this is great, and it's one way that you guys are combining the current technology with these historical records to learn more about our region, which is super cool. I know that there's some other really great tech around as well. And Amy actually asked, has there been study regarding changes in the sizes of some of the reptiles in the collection in comparison to the early samples? Do you mind talking a little about tech in that question?
Yeah, that's a really, really great question, Amy. Thinking about how we can use our collection that was maybe animals collected in the early 1900s to species or individuals that are collected now. And there's a researcher from SDSU named Dr. Rulon Clark who is interested in looking at those kind of patterns and questions. And he recently came in to collect toe samples off some lizards. And just like a tree ring has rings that you can tell, that the ages of that tree, our skeleton grows in kind of like growth spurts, at least for wild animals, they do. So he was looking at different growth rate patterns based off the toe rings, the skeletochronology it's called. So like the story told by the skeleton, and is looking at patterns of growth rates based on those toe rings. So it's pretty cool. And that's not necessarily size, but growth rate, I think, is something that's a little bit easier to discern, because sometimes, we might collect things that are smaller, and doesn't mean they're smaller altogether, but yeah.
Very cool. Any other sort of thoughts about the future of your field, where herpetology is going?
I think there's a lot of excitement around just like kind of thinking about how, let's see, kind of how we might use the technology in the future. So there's a lot of different unknown kind of aspects of what we might be using in the future, but there's, molecular biology techniques that are getting more and more detailed that will be able to answer questions about speciation and things like that, and describing different species as possibly being cryptic. So there's a lot of hope for using this information to also understand like patterns of decline and how to stop them and things like that. So yeah.
Well, this is fascinating, Frank. We are coming up on time, so I'm gonna just kind of pass you some audience questions rapid fire. I have some more of my own as well, but we'll start with these.